The overall goal of this procedure is to select Plasmodium falciparum parasites for binding to human brain endothelial cells. This is accomplished by first incubating a culture of Plasmodium falciparum parasites, which contain a mixture of infected and uninfected red blood cells with a layer of human brain endothelial cells. The next step is to wash off uninfected and unbound red blood cells by washing several times with culture medium. Fresh red blood cells are then added to allow further growth of the parasite population. The final step, a few weeks later, is to repeat the overall selection as soon as a sufficient parasitemia is reached. Ultimately, microscopy is used to show a high amount of parasites bound to HBEC5I.
This method can help answer key questions in the cerebral malaria field, such as the identification of parasite ligands or host receptor molecules involved in the cytoadherence process.
Prepare human red blood cells from group O positive whole blood by separating the red blood cells from the white blood cells by passage through a leukocyte depletion filter. Centrifuge the cells at 1000 G for 10 minutes and resuspend the pellet with rPMI incomplete medium. Repeat this wash step once more. Then, resuspend the pellet in rPMI incomplete medium at 50% hematocrit. Red blood cells are stored at 4 degrees Celsius for a week. Culture Plasmodium falciparum infected red blood cells with rPMI complete medium at 2% hematocrit and incubate at 37 degrees.